Say again? Ah, okay, okay.
Okay. Now,
Okay. So guys, let me introduce you to Dr. Bart Deplancke. He's a collaborator. We are working together in the construction of recombinant inbred lines in Aedes aegypti since 2015. So it's a long-term collaboration. And Bart, he's a very good researcher, a scientist, and he graduated as a bioengineer at Ghent University in Belgium in 1998. He did his PhD in immunobiology at the University of Illinois in the United States. And also he got a postdoc position at the University of Massachusetts in the United States. At the end of 2007, he became assistant professor in the Institute of Bioengineering in the School of Life Sciences at the EPFL. And then that's the short for École Polytechnique Fédérale de Lausanne. That's his institution in Lausanne, in Switzerland, where he established the Laboratory of System Biology and Genetics, and where he's since January 2020 full professor in this laboratory, since January 2020. And since September 2018, he is the Vice Dean of Innovation at the EPFL School of Life Science. I don't know if in 2015, he was also a guest professor of Ghent University and a member of the Swiss Institute of Bioinformatics. I don't know if he's still a member of these institutions now, because those are informations I got in 2015. Bart is also a co-founder of the software company Genome and co-founder of Alethea Genomic. That, I don't know, is it a software company as well?
No, I'll actually refer to it during my talk.
Sorry?
I'll refer to it during my talk, so you'll
okay, all right. So just to talk about his lab, initially his lab has focused on developing novel experimental and computational tools to improve the ability to elucidate the metazoan gene regulatory code and to detect naturally occurring genomic variation. Later, his lab developed research to understanding the regulatory mechanisms underlying fat cell development in diversity, as well as examining how variation in gene regulatory code affect phenotypic diversity. So his lab has therefore established itself as a group with international expertise in systems genetics and regulatory genomics. And Dr. Bart, he has lots of publications. He has published more than 60 or 70 peer-reviewed articles. I don't know exactly the number, but it's really a lot of papers he has published with more than 3,000 citations. So Bart, it's a big pleasure to listen to you and to learn more about Drosophila and how Drosophila is a wonderful tool for this kind of studies.
Thanks, Luis. Thanks for the very kind introduction. It's a very pleasure to talk to you all. I think those information, Luis, were still from 2015. I think since then, I think I have more than 100 papers, but you know,
really?
I was like, you know, I'm missing something. I think.
Yes.
Anyway, it doesn't matter. What matters is what I'll talk to you about for me this evening or for you this afternoon. Stop me if you have any questions or post something on the site, because let's make this interactive. Let's make this fun. Sure, I'll share the screen and, you know, let's see where it is. Now I lost it. Where is it? Okay, that's weird. Oh, is it again already? Wait. It worked beautifully the first time around, and now it doesn't. Let's see. Ah, there we go. Okay, it was missing for a bit. Okay. Okay, everything okay?
Yeah.
Yes.
All right. Okay. So as Louis alluded to, in my lab, we're actually primarily interested to understand how regulatory variation actually leads to complex traits. I hear some background, so perhaps somebody could.
You are not showing the
full screen.
The presentation screen.
Ah, okay.
You are showing
Is that better?
other window. Yes.
Okay. Now it's better.
Yeah.
All right. So you see my my cursor also moving?
Yeah.
All right. Okay, good. So as I said, we're sort of interested to understand how genetic determinants underlie complex traits. And I love to show this picture because it sort of makes the point that complex traits are really complex, right? Here you have a lady that reaches the noble age of 100 and is still smoking. So I think for many of us, this would be sort of an amazing achievement to actually challenge ourselves daily with cigarettes, but still actually reach the holy age of 100. And so the question, of course, remains whether this lady is just genetically robust or whether there's some magic formula underlying her genetic code that actually allows her to actually reach this age, despite actually the challenges that she submits herself into. And so actually Hippocrates already quite a number of years ago formulated this, I think, quite interesting statement that saying that disease is not a punishment from the gods, but is the outcome of environmental factors, diet and living habits. Therefore, we should use detailed observation as a basic for directing better health. And so I like the idea, of course, of using detailed observation. What Hippocrates was missing at the time, of course, is that genetics and the genome is actually important in defining complex traits. And so what he's actually saying here is that, yes, environment has a big impact, but if we now add the genetic dimension, we have to somehow then subtract the environment from this equation, because what we're really interested in the lab is how the genetics basically of an individual contribute to phenotypic variation, right? So we want to sort of contribute to Hippocrates' statement by adding this genetic dimension and then at the same time eliminating environmental interactions that actually might cloud our observations, okay? And so E often therefore acts as a confounder in studies aimed at understanding the contribution of G to a phenotype. And so how can we control for this? And this is how we actually arrived at the Drosophila genetic reference panel, which was originally started by Trudy McKay, who actually isolated Drosophila individuals from a fruit market, local fruit market in North Carolina. And then through sibling inbreeding actually generated a lot of isogenic lines, a little bit similar to what we're now doing with Lewis in terms of mosquito lines. Although here I should emphasize that these are independent, whereas with Lewis, we're actually starting from 4 founder lines here. These Drosophila lines are independent because they are derived from individuals that then were actually through sibling mating led to isogenicity through 20 generations, okay? So basically at the end of the day, then what we can use these lines for is basically different kinds of phenotypes, genomic sequence, and also we can look at molecular biology and gene expression changes, for example, okay? And so already a couple of years ago, what we actually were involved with Trudy McKay is to first of all actually genomically characterize all these 205 GGRP lines, okay? And so here I show you a fairly complex plot, but actually it's not that complicated in the sense that basically on the y-axis here, you have the number of variants, and on the x-axis, you have all the lines, okay? And so what you see is that basically you have several thousands of SNPs, so in a total of 580,000 SNPs all across those 205 lines. But in terms of genomic coverage, this is of course smaller than deletions and insertions that are of course large. Right? So even though the number of insertion and deletions are less than SNPs, as a whole, deletions and insertions, of course, affect more of the genome. And then you have these smaller deletions and insertions that were also mapped, giving in total actually a remarkable genetic variation per line. In fact, we sort of estimate that every nucleotide, every 10 nucleotides, there's sort of a genetic variant in those lines, right? If you now put that in perspective in terms of human variation, it's sort of 10 to 100-fold greater than what we see in human. So we always joke in the lab as to how Drosophila individuals must look at one another. If we look at each other, we see sort of 1 in 1,000 base pair genetic variation. Drosophila sees 1 in 10. So actually the phenotypic variation must be remarkable. And I'll show you some of that because we've then actually used this resource exactly to study phenotypic variation. Okay. Now, one component that is sort of missing and that we actually contributed to is, of course, mitochondrial genomic variation, which is often overlooked even in human genome phenotype or genophenotype relationship studies, even though basically we now know that the mitochondrial genomic variation is actually linked to a slew of diseases, many of them actually quite serious, going from neuropathies to heart conditions, eye neuropathies, liver hepatopathies, and so forth and so forth. So mitochondrial genomic variation does matter. And here we actually have a unique opportunity to look at mitochondrial genomic variation, especially because, as I already indicated, the lines are independent. So the mother actually is contributing the mitochondrial DNA independently for each line. So this is actually then a remarkable resource also to study mitochondrial genomic variation. So I'm not going to go into the details of how we then isolated basically mitochondrial DNA specifically in each of those lines, just to sort of mention that GGRP lines are genetically independent, but limited mitochondrial coverage so far. So we had to actually devise a system to actually specifically isolate mitochondrial DNA. Ultimately, we were able to do that. And this is sort of the result of that. So here you see actually the circular mitochondrial genome, which is actually not all that big. It's sort of around 20 kb in total, right? And what you basically see here is basically the gene. It's very gene dense for all the genes that are basically encoded by the mitochondrial genomic DNA. And then I sort of plotted here actually all the different variants. Many of them are synonymous. So the SNPs, they don't affect actually the coding function of the genes. There's about 241 SNPs in total. We found 15 indels actually in some interesting genes, and then multinucleotide polymorphisms were found as well. Just to say that most of the variants actually seem to happen actually sort of in a low allelic frequency. So they are sort of singular or at least unique to only a few lines and not actually very abundant. And that allowed us actually then in general to focus on common genetic variation in the mitochondrial DNA, and then to partition actually the DGRPs in sort of 13 haplotypes, in which these haplotypes basically, in which the DGRPs for each of those haplotypes are sharing more or less the same mitochondrial DNA, okay? And so for the first time now, what we could do is to use these 13 haplotypes, which basically covered around 150 of these DGRPs. So basically each haplotype had roughly sort of 10 DGRP lines. What we can now do is actually associate these different haplotypes with different types of phenotypes, okay, that were already mapped in the Drosophila DGRP panel. And one of them was actually very interesting because when we actually, for example, looked at this one paper here, which actually measured food intake, in this case in males, what we discovered is that some of those haplotypes had significantly higher food intake than some others, for example, this one, right? So in total, we actually were able to correlate mitochondrial variation or DNA variation with about 259 non-correlated phenotypes, which already had been measured in the DGRP, okay? And 12 phenotypes passed, especially those related to metabolism and stress response. And one of the remarkable ones was actually the one in food intake that I just mentioned, right? In which
Mark,
this particular
can I ask you
haplotype.
one thing? Can I
Yeah,
ask,
sure.
In this graph, are you showing the different lines?
So yes, let me clarify this. So each of those box plots, so to speak, is one haplotype, and the little dots in each of the box plots is basically one DGRP line. Okay.
Ah, okay. So you have 13 these haplotype. Okay.
Yeah, exactly. So all the DGRPs are sort of partitioned according to the haplotypes. Okay. And if you then measure basically the average food intake per line and you partition that per haplotype, you can see that all the DGRP lines that belong to haplotype, mitochondrial haplotype 5, had a significantly higher intake than those actually in haplotype 1, which had a sort of lower intake as measured sort of with this capillary assay. Okay.
And how do you measure food intake?
So this capillary, so you actually, we have this assay set up and you have a very fine capillary that actually allows you to measure very fine volumes of liquid. Then you let the flies basically on the food, so to speak, which is sort of liquid food. And then you can see actually the difference in food intake through this capillary in terms of overall volume that were actually decreased in the capillary. So that allows you to actually, in a very fine-grained manner, actually measure how much food intake over a specific amount of time flies actually can digest, right? So
but that is influenced by the type of food.
Of course. Here, I mean, all the lines were subjected to exactly the same amount, the same type of food, of course, right? So we're trying to minimize any kind of variation across the haplotypes in that the food was basically, consistent across the haplotypes and measured in exactly the same fashion at exactly the same time, because I'm going to, of course, show you later that actually haplotype and circadian time, of course, matters in the flies as well, right? Now, this was not our data. We actually just took opportunity from the fact that there's a large community out there that measures phenotypes in DGRP lines, right? And so we just actually took the data from this paper by Garlapov, which allowed us to actually find this very interesting difference in food intake between at least two haplotypes, right? And, I'm going to spare you the details because I want to spend a little bit more time on another study, but you can sort of read this up in the respective publication. But interestingly enough, when we actually took some of those lines, 5 and 1, and then we swapped the mitochondrial DNA, which you can do through the crossing, right? What we were able to show is that by swapping the mitochondrial DNA in those lines, that we were actually able to reverse the food intake. So if lines actually with mitochondrial haplotype 1 now receive haplotype 5, they now actually starting to eat more and vice versa actually for haplotype 5. Indicating that indeed mitochondrial DNA and the underlying variation actually can affect complex behavior in this case actually food intake. I'm sorry?
Someone has the microphone open. Please turn off the microphone, people.
Or was there a question perhaps? I don't know. There was no question. Okay, good. So we thought that was a quite remarkable, and it shows you, I'm just sort of showing you a little bit the tools we're actually able to gather now for these DGRPs, going from nuclear DNA all the way to mitochondrial DNA. We can also look at interactions. And this was, for us, one very interesting phenotype that now we could clearly actually associate to mitochondrial DNA, which, as I already indicated, is often overlooked, okay? But what I wanted to spend the most time on, and this is also sort of with reference
just
to,
one more question about
sure.
that set of data. This difference is behavioral or metabolic? Do you know? So it just eat more, or it has a higher energy expenditure and then becomes hunger?
I think this is a very interesting question. To be completely honest, I don't think I can fully address it in a sense that, of course, the mitochondrial swapping was not tissue specific. So it could also have affected neuronal activity, which might then be more behavior versus basically overall metabolism. So that, of course, we couldn't actually distinguish. So I think it's a very good question. And, I think the only way to sort of do this would be to do this sort of in a tissue specific fashion, such that you can really see what the contribution is of each tissue. But I think this is sort of very hard to do. I wouldn't actually immediately know how to actually do this tissue specifically. But it is a very good point. So far, I can't really distinguish between those scenarios. But I think it's an interesting one. And it sort of brings us actually to the next topic. I think if there's no more questions. No.
No.
Which basically, yeah, sure.
Okay, it's okay.
It's okay. Okay, yeah. So it brings us to the next topic, which is actually a sort of a tribute to Antonio Mireles Filho, who some of you may still know. Is actually is acquainted to Luis, and who came to my lab as a postdoc and was responsible for actually making the link between the Fiocruz Institute and our institute. So I'm very still grateful to Antonio. So I thought actually I spend most of the time on actually telling his story today. Which was accomplished by Antonio together with also a fantastic PhD student who already graduated now, Masha Litovchenko, who actually did most of the computation analysis that I'm going to talk to you about. And so again, the idea here is quite simple, is basically how can we now link genetics with a complex phenotype, which is basically circadian behavior, okay? And we all know that basically there's different kinds of circadian phenotypes out there, even in humans. You have those basically that are, morning people who are very active, but those that cannot get out of bed. And then you have those that want to go to bed immediately, and then, some others that actually want to go through the night, right? And so, these are the morning and the evening people, but in the meantime, many more phenotypes actually have been mapped. For example, I'm more of a napper in the afternoon. I like to nap, but I'm both a morning and an evening person. So there's different kinds of phenotypes related to circadian behavior. And of course, one of the question is, in how much is that genetically encoded, right? And Drosophila, of course, is a fairly interesting model to study this sort of question, because, of course, in 2017, a Nobel Prize was given for discovering basically the roadmap of how the circadian clock works. Because there's the clock and cycle and the period and the timeless regulators basically who form this negative regulatory loop, and then as such basically start to produce these oscillations. Okay. And so one of the questions, of course, that we wanted to ask is, first of all, can we set up the circadian clock? Can we study it actually in quite some detail and ask ourselves how many genes are expressed in a circadian matter, and can we identify novel master regulators of this particular circadian clock? Okay. And so what Antonio and Masha actually set out to do is to, first of all, actually use white mice, sort of the workhorse of Drosophila research, and simply actually try to map circadian gene expression, this oscillatory pattern that is actually visible right here, in flies in 4 different tissues: the brain, the fat body, the gut, and the Malpighian tubes. Okay. And the idea here was to actually collect flies every 2 hours for 48 hours, first in a light dark pattern. And then just to make sure that we're actually detecting true circadian behavior, we had then the next sampling is actually happening in a dark pattern, right? Just to make sure that basically we're not just looking at light entrainment, but truly actually circadian gene expression, okay? And so this is sort of a total of 192 transcriptomes that we actually envisioned having to profile, which is, as you probably can imagine, quite a bit. And this is actually where, I'm coming back to Lewis's initial point, where basically a couple of years ago, we generated a sort of new way of doing transcriptomic profiling, which was actually based on the principle of single cell transcriptomics, in that we actually very early in the process of reverse transcription add a barcode to the cDNA, such that basically already at a very early step during second strand synthesis in making the cDNA, we can pool together already all the samples in which we were interested to profile, okay? And afterwards, we proceed with just the tagmentation step with the TN5, which is now often used in single cell transcriptomics as well. Then we index the library, amplify, and then we sequence, okay? And so we call this tool bulk RNA barcoding and sequencing, okay? And the true advantage of this tool is that it's virtually as good as the classical workhorse, which you may be familiar with, which is TrueSeq, in that, we actually found a very good correlation actually in overall gene expression, in number of detected genes, number of overlapping differential expressed genes. But the true difference is basically the cost, whereas TrueSeq is typically, I don't know how much you would pay, for example, in Brazil, but here, we still pay actually over 50 euros or dollars actually per sample. Whereas if you actually use the BRB-seq protocol, it actually comes down to a couple of dollars in total to actually make your transcriptome. And so at the end of the day, this is cheap, very easy to do, and equivalent to sort of 4 gene measurements by qPCR. So, or
Mark, sorry, can I ask you
Yeah.
one thing? Is this the same technique we will apply in Aedes aegypti for the single cell sequencing, or something different?
It's sort of similar, but we optimized it in similar in several ways as to how much RNA is needed, which RT reverse transcriptase actually was working the best, which barcodes are actually working together seamlessly, because not all barcodes are compatible, and so forth and so forth. So there's a couple of optimizations that one had to do, but it's definitely not rocket science, if that is what you mean. I mean, it's basically sort of an early barcoding step that allows you to actually profile in a very cheap fashion, actually many transcriptomes at the same time. Okay.
So at the end, just a small technical detail. How much reads you have for each sample using that approach? How it doesn't get limiting?
Yeah, so basically what we see is, so this is something I should have mentioned also. So this is basically a 3' end transcriptomic profiling technique. So if you're interested in, for example, splicing, then this is not for you. But most of the applications, we would argue, is basically simply transcript counting, okay? And for that, the 3' end is more than sufficient, okay? So if you actually limit, so to come to your question, if you then limit basically the full transcriptome coverage to actually only the 3' end, which is only 100 base pairs, you can vastly actually reduce the coverage you need in order to actually quantitatively count the expression of all of those genes. So at the end of the day, we sort of routinely get 5 million reads per sample, okay? So if you do actually, you know, a next gen, one of these next-seq runs, and now you already have NovaSeqs, which have become routine, but even the next-seq, you can typically actually do easily up to 100 actually, samples, especially Drosophila, because the transcriptome is even smaller than that of human and mouse. So we actually do, in this next-seq, we do easily actually 96 to even 120 or so samples in one run, okay? And so that actually vastly reduces the cost overall, because then the cost for making the library is actually almost nothing. It's basically the sequencing that will be the final cost at the end of the day, right? So our estimate is now that with the sequencing, we can sort of do transcriptomes for roughly basically $20 to $25 a transcriptome, okay? So that actually makes things very affordable. And truth was, you know, we had no intention to basically make this commercial because we just thought this is sort of cool for the community. But we had so many requests by all over the world to actually help them with the BRB-seq protocol and how to do this, that actually the people, and then here that especially Daniel, who actually developed this, has now actually founded a new company, which is Alethea Genomics, and basically is now selling them as kits. So it's actually quite easy. We sell it, you know, we send you the kits, and you can do the BRB-seq right in your lab, you know, without any problem. And it's much cheaper basically than most commercial applications out there. So I'm a co-founder of the company, so I don't want to sort of commercialize it too much here, but at least it should democratize. That's a little bit our intention, transcriptomics tremendously, because the overall cost now is really becoming affordable, especially for large-scale projects that you might have in mind. Okay. So I sort of introduced it because now you sort of understand how we got to the 192 samples that we needed in order to actually profile circadian gene expression in the GRP line. So all of the data here was actually generated with the BRB-seq protocol. Okay. And just as sort of a benchmarking, here I show you basically timeless, which, as you know, probably is a central component of the circadian clock. And I show you basically now the sampling over the 2 hours, you know, in 48-hour time. And what you see here is normalized expression. So you can nicely see basically these nice oscillations of timeless in the 4 different tissues, right? So this works actually quite well. And so what we can do now is actually look at how many other genes actually we find cycling. And what you could see basically is that, first of all, you have quite a few genes that actually only cycle in one tissue, right? So for example, here 479 in the gut, 135 in the brain, 359 in the fat body, and so forth. But you also have 14 genes basically that cycle in all tissues, okay? And so we sort of looked at those and reasoned that those might be master regulators. And sure enough, actually, many of the 14 were actually already core circadian genes that I already alluded to in the beginning of this topic, like period, timeless, cry, and so forth and so forth. So this again validated sort of the overall approach. Not only that, but here I'm only listing sort of 7 of the core ones, but what about the others, right? And so here you basically see quite a few others actually that show quite a robust cycling pattern actually across the tissues. I mean, of course, timeless is probably always the neatest example because it's sort of the most robust, but some others actually are clearly cycling also quite well across the tissues. And so what we did actually together with, together with Antonio, what we did is actually used drivers and the promoter of timeless actually to knock down those new master regulators that so far were actually not proven to be involved in the circadian clock. And so, we did RNAi-mediated knockdown in timeless positive cells. So those neurons basically that are typically driving arrhythmic activity of 4 candidate genes that were sort of novel. And what you can clearly see is that knockdown actually compared to the control clearly actually reduced the percentage of flies that actually showed rhythmic behavior. Okay. The period was actually not affected in that most of it was sort of around 24 hours. But you can also see that the rhythmicity index was actually slightly lower as well. But the most significant effect was actually on the number of flies that actually was ultimately rhythmic, sort of suggesting that some of those genes are clearly sort of quite important in the circadian clock and allowed us basically to map new core regulators that we can then actually study in a little bit more detail. Okay, but I wanted to tell you this story not necessarily to really go into detail.
May I make a small question? So
sure.
You would not expect a more pronounced loss of rhythmicity, because still 60% keep being rhythmic.
Absolutely. So this is a very good point. I cannot see actually, but even if you actually knock down some of the core regulators like clock and so forth, the overall decrease in rhythmicity is actually also not that striking. So what probably is actually happening is that the knockdown is often not complete. So you sometimes have actually incomplete penetrance. And as a result of that, basically, you still have flies basically that sort of survive this knockdown and actually show more robust rhythmicity. It might, of course, also be that they have sort of more peripheral roles in this core circadian clock, such that basically there might be some redundancy for some of those genes that allows those flies actually to still show rhythmic behavior. So there might be a couple of reasons why the percentage decrease is actually not absolute, let's put it that way. But overall, I would say this is sort of not entirely unexpected, because even some of the core genes, when you try to knock them down, you still actually see quite a bit of flies that show rhythmic behavior. And that, of course, also has to do with the fact that entrainment is really robust. I mean, they're really sitting in these incubators with light and dark patterns, which are really very, very solid and very regimented. And so as a result, basically, you might not even necessarily need the circadian clock to still show rhythmicity. So that might also be sort of an influence. Okay. I hope that answered your question. Yeah. So what I really wanted to actually allude to in this story, and this goes back to the influence of genetic variation, is this very exciting observation, which actually already dates from about 22 years ago here, in which basically they show you these rhythmograms. There's these plots that actually show you nicely the rhythmic behavior of flies. So here you see the dark period, and then around the switch between dark and light, you can see basically quite a bit of activity of the flies, so indicating that they're sort of waking up and become active. And then sort of activity is sort of more moderate, and then right before actually the light will switch off again, you see actually quite a bit of behavior of activity. Okay, so that is very typical across 2 different strains, Cantonese and Oregon. You can see sort of this nice pattern. But what is really interesting is when you actually allow those flies to live in a complete dark regiment. Okay, what you can see is when you actually switch off the lights completely, is that the circadian clock starts to shift, right? So here you can see that basically the flies are confused and they start to actually become more active earlier, but also longer. Okay, so it's almost like their circadian clock sort of increases in period. Now in Oregon, you can see the opposite. So the flies are again confused and they anticipate sort of the light arrival much sooner, even though the light will never come on. So you can see this shift actually to the left, which sort of suggests that the period is also getting shorter, right? So the fact that these are 2 different strains sort of suggests that genetic variants might actually drive this very interesting actually behavior. And so this is what we really wanted to investigate. How is this observed phenotypic variation in behavior, physiology, and metabolism encoded at the genetic molecular level? Okay. And so this was the question. Can we now use again this panel of DGRP lines in order to go from genome to phenotype, right? Okay. So we already have the genomic variation. I already talked about this plot earlier in my talk. We already mapped the genetic variants, but how do you now actually measure circadian behavior in individual tissues, right? This is, of course, a much more difficult. Now, we thought that actually we could infer circadian time actually by measuring gene expression in the individual tissues and using, for example, some of those core clock genes as our guide, because we can sort of, based on the oscillatory pattern of timeless, for example, infer sort of where the circadian time is or at which circadian time we are in individual tissues, okay? But if you then think about it, this is truly actually a remarkable challenge because we would have 140 lines, 4 different tissues, and then we would have to measure basically for each of those lines every 2 hours gene expression across 48 hours, right? So this is a couple of thousand transcriptomes, which, you know, is simply too much. You know, we could not actually sort of think about doing that, but rather what we actually used is sort of an alternative, cheaper and a little bit a cheeky strategy, but that actually ended up working quite well. So the idea here was simply that rather than actually sampling like we did for white minus across 48 hours every 2 hours, what we did is actually we divided basically now 24 hours here, and then in basically divided by the 140 lines that we actually decided to look at. And the idea was to simply actually measure then every roughly 8 or 9 minutes, one line across, and here we chose actually 3 different tissues: fat, brain, and the gut, okay? So rather than actually doing this individually across many time points in each line, now we measured basically in each line only once, one tissue, 3 different tissues per line per time point. So for one time point. So basically, as you can see here, for example, this one was DGRP 21. Then 9 minutes later, we took DGRP 22, 23, 24, and so forth, all the way such that we can now actually measure all 140 lines across this circadian window, okay? So this is sort of a key slide in order to understand our experimental setup. But the good news now was that basically, rather than actually having thousands of transcriptomes, now we actually had to do basically 140 lines times 3 tissues times, which is basically sort of 520 and a little bit transcriptomes. So at the end, we sort of had 4028 that actually truly worked and allowed us to map the different gene expression profiles basically in those tissues across all the lines, right? And if you now were able to follow a little bit, what was really remarkable is that overall, we could actually nicely reconstitute, for example, here, the expression of timeless. So let me guide you through this plot again. So this is light, this is dark. And each dot is basically the expression level of timeless in one particular DGRP line, okay? So basically in total here, you see 140 dots, which means 140 DGRP lines, okay? And this is normalized count. So it gives you the expression level of timeless basically across these 140 different lines across the light and dark regimens, okay? So most of them actually followed this pattern quite nicely, but you can immediately already see that some of those lines actually don't follow the expected pattern. So we call these the outliers, which you can actually nicely see here in orange, okay? I hope that was clear, because now what we can do is, first of all, actually train on the white and some other DGRPs that we actually did profile all across the light and dark regimens. So it's about 5 of them in total, okay? And we use that as a training set, first of all, to decide which half of the day a sample is actually derived from, whether it's actually from the light or the dark, because since this is oscillatory and cyclic, you might not be able to differentiate actually between the light and dark patterns, okay? And then basically we had to use another sort of classifier to then decide what the physiological time is of a specific sample, okay? So for example, what we try to do is to simply do some machine learning to try to find out at which circadian time this guy is specifically, right? So that's a little bit the exercise that we're trying to do. So for every DGRP sample now, we had to infer half of the day with with sort of this classifier. It's a lasso regression for those who are interested. And then the predicted physiological time and compare with the time of harvesting, which gives us then a difference, in terms of inferred time versus the time in which we actually sample that line, which then gives you sort of a shift for the circadian time for that particular outlier. Okay, this all sounds a bit abstract, but hopefully it's going to become clear quite soon. Okay, so here I'm sort of summarizing all the data in the sense that all of those lines basically were found to be outliers in our data. Okay, and what you can see, for example, if I now focus, for example, on this guy here, 2081 or 28,172, which is just the DGRP number. Okay, you can see basically here that the difference between the physiological and the harvesting time is roughly 8 hours. So that means basically that even though, for example, for most of the lines, for example, now it's noon, right? This guy basically could be either at 4 o'clock in the morning or at 8 o'clock in the evening, because the difference is overall 8 hours between what would have been expected and what we actually inferred from the gene expression data. Okay, so that's basically, if there's anything you want to remember from what I just told you about, what we just did is to be able to actually count the difference or calculate the difference between the inferred physiological time based on the gene expression data and the harvesting time. So for example, if we harvested this line at noon and we see basically a physiological time of either 4 hours in the morning or 8 o'clock in the evening, then there's a clear difference of 8 hours that must be caused by genetic variants, okay? Because all the rest is sort of the same. And what you can see basically is that most of the shifts actually occurred only in one tissue, which is already very interesting. So basically, what means that overall circadian time in the flies is synchronized across the tissues, except that because of genetic variation, sometimes one or multiple tissues, like in this guy here, are desynchronized. Okay. For example, here, this guy had actually circadian variation, both in the fat body and the gut. And this guy actually had circadian variation, both in the fat body, gut, and the brain. Okay. So extensive molecular variation, mostly in a single tissue, and genetic determinants therefore act mostly at a tissue-specific level. Okay. Now, I point out this guy because basically this is very interesting. This guy actually presents strong differences in all tissues compared to the expected physiological time. So clearly something is wrong in terms of circadian gene expression in this particular line. So we were interested in actually finding out what exactly is going on. Okay. So what I'm showing you here basically is sort of the wild type or the white minus reference, right? And now we did exactly the same thing. So we took this line and now we actually profiled this line again across the basically light and then dark regimens, okay? And so if nothing was going on, we would expect exactly the same sort of oscillatory pattern as we would see in the control line, okay? But what we saw actually was, for example, if we contrast the brain and gut timeless expression profiles from the white minus to the DGRP 2833, which is the one that showed some remarkable variation, you can see actually that the oscillatory pattern is almost absent, okay? There's almost no oscillation going on for timeless, which suggests basically that this line, circadian clock, is somehow broken, okay? Not only that, if you now actually looked at gene expression rhythmicity across the different tissues, what you can clearly see is that you have much less genes that are rhythmic. And in fact, none of the genes, we found no genes that basically were rhythmic across all the different tissues in that one line. So this line has undetectable rhythms in clock genes and dampened rhythms genome-wide, because as you can see, if you now actually calculate the amplitude of the gene expression, which is basically, if I now back up a little bit, is simply the difference here between the peak of expression and sort of the valley of expression. So this is sort of allowing you to calculate the amplitude. If I now plot this for this particular line, what you can clearly see is that the overall amplitude is actually much lower, which in fact was already visible right here, because you can see that the overall amplitude is much lower than what it is actually in the control line, right? So clearly the circadian clock is basically broken, okay?
So you showed gene expression, but they oh, okay, that's it. You
Yes.
show it. But
So now I'm actually showing you activity, not only gene expression anymore. So this was a timely question. So now we actually show again locomotor activity basically across the dark and light regimens. And this is basically for the white minus. And again, you can see this sort of nice activity in anticipation of the emergence of the light. And the same thing when actually the light will go off, again, you see in this activity. And interestingly, basically, what we could clearly see is that basically the same thing actually holds true, even though the profiles are perhaps a bit smoother in the 2A233. So this is quite remarkable. In terms of expression profile, this fly line actually has hardly any rhythmicity in terms of gene expression. But in terms of behavior, it actually behaves exactly the same as the control line in terms of overall circadian behavior. So this poses a little bit of
that's not the effect of environments. They are not being trained by the light-dark cycles. I mean, what you can see in the GRP 28233 is just a response to the turning on and turning off the lights, response to the lights.
Well, let's find out, Luis. Okay, let's find out. So what we did actually is we actually did a little trick. So what we did, we then subjected those same lines again to the dark, complete dark, except that now here we basically provided a light pulse at CT15, right? Which disturbs, of course, control lines tremendously because they pick up this light and they sort of adjust to this new light stimulus. So what you immediately see is this shift, okay, in overall circadian behavior, in that they're trying to adapt to this new light stimulus that they sense basically in the dark regimen, okay? So you can clearly see in the control lines that they adapt to this light stimulus. Now, interestingly, what you can see for the DGRP 28233, even though we provided the same kind of light pulse, they're not adjusting basically to this light pulse. So something is wrong. They show rhythmic behavior, but they are not able to pick up this light pulse. Okay. Does that make sense, Luis?
Uh-huh.
Yeah. So what is going on? Okay. So that was a big question. And here, Antonio, of course, used his great genetic skills in Drosophila. What we're now trying to do is to see if we could pick up an important mutation in that line that could explain this sort of remarkable behavior. Okay. And what he did was actually chromosomal mapping, right? In which basically he, one by one, replaced basically single chromosomes from this one DGRP line into the white minus to see if he could recapitulate the phenotype. Okay. And what you can see is that only when you actually, let's first look at the initial situation where basically here you provide the light pulse, and now you see in the control lines this phase shift. Okay. So it immediately adapts to this new light pulse and this new regimen, so to speak. Whereas the DGRP, the one that I already talked about now for quite a while, did not actually respond to this light pulse. Okay. So the idea was to find perhaps one chromosome that allowed us to recapitulate this particular phenotype. And what you can see is basically that the second and the X chromosomes do not allow you to actually recapitulate the phenotype, but the third chromosome does. Okay. So something on the third chromosome, a genetic variant likely, is actually responsible for this kind of phenotypic behavior. So we look back again at all the list of genes that we actually could think of and look at, for example, important circadian genes. And sure enough, actually, when we then looked into CRY, okay, what we actually discovered, we have to be honest, initially we already had seen this deletion, but it didn't actually seem to actually cause a significant loss of function. It sort of was sort of a synonymous deletion. Okay. Well, actually, it was a longer deletion, but actually, according to our phenotype impact assessment, it didn't really actually cause a remarkable. It shouldn't have caused a remarkable phenotype. So we were sort of puzzled. But let me just actually focus on this. So here you see this actually small deletion in this one line in the cry gene, one of the core circadian clock genes. And what actually happens is that this methionine and tryptophan is sort of deleted, and then we have sort of a small, interchange of amino acids down the road. Okay, so the question now simply is that sufficient to cause this particular phenotype, right? So here we got lucky because we actually worked together with a colleague of ours here at EPFL, Matteo Del Perraro, who is an exceptional structural biologist. And what he did is to actually use the reference cry protein structure from Drosophila and map basically the DGRP2A2333 amino acid sequence or structure on top of that. And sort of to our disappointment, we did not see any change, which is sort of what I meant to say when we actually had done an initial sort of genophenotypic assessment. Okay, so what is actually going on? And then luckily, Matteo, my colleague, actually looked a little bit closer and actually looked at these different tryptophans, which actually are responsible for the cry, the cryptochrome, so to actually respond.
Só agora que entrou monte de coisa, uma coisa atrás da outra, e...
Okay, someone was with the microphone on. Sorry.
Yeah, no problem. So basically, these are the tryptophans that are in cry, are responsible for the electron cascade, which allows basically individuals, Drosophila in this case, to respond to light. Okay, so these tryptophans need to be arranged in a very specific fashion in order for those electrons to jump from one tryptophan to the next. Okay, and what was really remarkable is that even the alpha helix basically of the mutated form was actually completely matching. What was actually remarkable is that one of those tryptophans, 420, was no longer actually at the correct position, right? So it sort of shifts on the other side of the alpha helix from here to there, which makes us now hypothesize, and this is probably the first in vivo evidence for this electron cascade actually, is that this electron cascade in this mutant line is no longer happening because of this very small deletion, which actually slings the tryptophan that is important to initiate this cascade actually on the wrong side of the helix. Okay, so long story short, what we actually discovered and generated is a rich data set for circadian biology with high resolution and more than 700 transcriptomes. We found new putative clock genes, and we found a naturally occurring fly line. So keep in mind that this is a natural genetic variation. So this mutation is actually stratifying somewhere or segregating somewhere in nature, containing a new cry allele with a night locomotor activity pattern and disrupted molecular clocks. Okay.
So Bart, the cry, if I remember well, the cry is a photoreceptor.
Exactly.
The photoreceptor, and then it changes conformation when the light's on, so that it can activate the circadian loop. So this change in tryptophan is making the conformation not to happen at all?
No, it's what I meant to say is that basically the conformation is correct. It's just that basically the electron cascade that actually needs to happen, or it has been hypothesized to happen, because it's very difficult to actually demonstrate this truly, but through mutations, basically, we've been able to discover the true importance of those tryptophans. So they're key and they're highly conserved, as you can actually see here. Just 1 second. So here you have all those tryptophans. So here at 397, you have 420. You can see the W's basically nicely aligning. You know, what is actually really hypothesized is that this cascade here is crucial for the photoreceptivity of this photoreceptor, right? And there might be conformational change, but this is sort of the activated form of cry in which those tryptophans become aligned, right? And so if you have the mutation, those tryptophans are no longer aligned, which is, I think, very interesting. Just to think that basically this very small sort of hiccup, which actually just throws the tryptophan on the other side of the helix, has this dramatic impact basically on the overall phenotype. Except that, as you said, Luis, and this is of course true, right? If you now actually allow me to go back, because of this very harsh light entrainment, which you alluded to, Luis, right, from the light dark, they're actually from afar, nothing is wrong with them. They are actually able to follow sort of the light-dark cycle, and they're anticipating it correctly as the control lines would do, except that they would have no ability to adapt to a new light-dark regimen because they're simply unresponsive to basically changing light patterns. They're sort of blind, so to speak, for this basically light entrainment, which I think is a very interesting phenomenon, right? So the circadian behavior from at first sight is completely correct, but then if you actually try to perturb it, they cannot respond to it. Okay. And so this is a naturally segregating variant, right? So this is picked up actually through inbreeding, but it means that one of those individuals must have had this variant from the beginning, right? And so sort of the take-home message of this entire story, which I find quite intriguing, is that basically through this study now, we've been able basically to sort of demonstrate that the circadian clock is not always completely synchronized throughout the body, in that certain tissues may have actually an altered circadian pattern, which is in a way desynchronized with the rest of the organism, so to speak, right? So what I mean with that is that, for example, here you may have the brain time, gut time, and liver time, which sort of is the control line in most of the individuals. Then in this cry mutation, you may have something completely weird. But then in some individuals, for example, your brain and liver time may be correct, but your gut time may actually be shifted, right? And we've been speculating and having a lot of fun in the lab with that because, for example, many people, some actually really like a lot of breakfast and some others don't. They are like, oh no, I'm just going to have a coffee and then actually continue my life and have something bigger actually down the road. And it sort of would be, it's very interesting to speculate that actually the circadian activity of your individual organs might actually not be synchronized. And so you might be awake, but your gut is still not awake, right? And that I hope to have conveyed to you, at least in sort of a semi-convincing manner, although it's a complex study to grasp completely. I hope I sort of conveyed this idea, but this is what I meant here, right? So that, you know, for example, in those lines, the gut is actually desynchronized from the fat body and the brain. So the individual, this fly individual might have an active fat body, might have an active brain, but the gut has not been synchronized yet, right? And I wonder actually to what extent, of course, this applies also to humans. It's much more difficult to study, but you know, this study in flies suggests that this might actually be much more prevalent than we may anticipate. Okay, so that's a little bit the take-home message of this, which I find quite intriguing. It's of course a little bit hand-waving and speculative, but I find it nonetheless actually quite interesting. And so, if I have 5 more minutes, Luis, would that be fine?
Yes.
Yeah.
That's cool.
So I quickly want to finish actually, because in my title, I did not only have phenotypic diversity, I also had cellular diversity, okay? And, you know, we can study all of those phenotypes to the greatest detail, but of course, if you don't know actually which cells your organism of interest is made up of, then how can you actually understand any type of phenotypic behavior or phenotypic variation, right? And so to illustrate that, of course, and this will be very trivial to all of you, but nonetheless, to sort of recapitulate that a little bit, it's sort of similar to this idea of going to your garage with your car saying, I have a noise problem, and your garage or the technician at the garage saying, well, I don't know where this noise might be coming from because I have no idea what is in your car, right? And of course, we know that your car consists of thousands of different pieces that all might make noise, right? So you have to sort of try to map all the different, you know, elements of your car. And similarly, in your organism of interest, you need to be able to actually find all the different elements, which is the cells and the cell types that make up your organism. And it's sort of, you know, very interesting to have to convey that basically even today for human, mouse, or Drosophila, we've still yet to map and understand all the cell types that are available in our organism, right? And so giving our love for Drosophila and our interest in the lab to do single cell transcriptomics and to make new technologies, you know, about 4 or 5 years ago, we sort of got a consortium together with the specific goal of trying to map all the cell types in Drosophila. We've of course failed so far. We've done our best. I'm sure there's still more cell types to be had, but at least we were able to provide a first reference map. And so this has just been actually published in Science in 2022, you know, in the beginning of March. And I just want to finish my talk a little bit with sort of highlighting a couple of things, not going into any detail because you can read the paper, but just to sort of give you a grasp of what is out there, right? So we took an adult fly, no conditions so far at this point. We dissected 17 tissues, profiled the transcriptome of 580,000 cells, and, you know, through actually a very active and very fun sort of jamboree-like annotation system that involved all sort of experts in Drosophila biology, we were able to annotate all of these cells and actually sort of identify so far more than 250 different cell types in the fly, okay? And what is really cool, and I think really will speak to this consortium here, or the community, is that cells are everywhere, and no better organism to show that is insects, right? And why? Because what you can do with insects is to simply take, for example, a head, take a body, grind those individual body parts to pieces, and try to actually grasp and profile every cell that is part of that body or head. And why is that so remarkable is something we cannot do in mouse and human, obviously, is illustrated in the following slide. So here you see actually all the cells stratified according to different tissues, you know, testes, antenna, ovary, fat body, and the different colors. I'm not going to go through all the detail. But what I want to emphasize here is the following. So what you see here basically is in red, and I'm not going to make it very complicated, basically in red are all cells basically that would not have been discovered if we only would have stuck to these 17 known tissues that we dissected. In other words, if you grind the entire body and the entire head, you find actually cell types that we don't even know where they are because they don't belong to any of the canonical tissues, right? Which I think is fantastic, right? So by actually grinding the entire animal at once and trying to grab all the cells, we can actually map clusters of cells that would not have been visible if we would only have dissected individual tissues, right? So they must be part of the cuticle, part of other parts of the body that we have no way of really actually dissecting, right? So I think this is already one remarkable thing of the flies. And so if you're interested in looking at this data, we've actually generated 2 portals that would really allow you in a sort of simple fashion to look for your favorite genes, look for your favorite cell types, see which genes are expressed there, see which genes might be differentially expressed from one tissue to the next, and so forth. We've also done cross-tissue analyses. For example, hemocytes are present in all individual tissues, yet they have different gene expression profiles, a little bit similar to like how macrophages, for example, in human and mouse are present in most tissues, yet have very different gene expression profiles according to the tissue that they're coming from. We found actually quite remarkable sexual dimorphism because we profiled both males and females. And so not all cell types actually, even in shared tissues, are the same. We did trajectory analyses that allowed us actually to have new insights in how certain organs develop. And we also then actually looked for gene regulatory networks that might be responsible for these differentiation programs, okay? And why do I mention all that? Because now one of the ideas is to, of course, use the resource that Louis is building very nicely at Firecruise to do something similar, but now for mosquito, right? We now are in a fantastic position to actually build similar types of atlases for different types of insects. And I think for many reasons, of course, the mosquito is of great societal interest. And so here again, we have the fortune not only to be able to work with Firecruise, but also with the BioHub at San Francisco with Steve Quake, funded by the Chan Zuckerberg Foundation. And so we're very grateful then to Robert and Max and Vincent, who are helping this in this fantastic endeavor. So this is yet to come. So if you want to have me back in maybe 1 year or 2 years or so, hopefully then we have a fantastic map of the mosquito ready for you, which I'd be happy then to talk to you in great detail, but maybe Louis can do this for you as well. All right, so with that, I want to thank you for your attention. I hope I didn't actually spend too much of your time. The collaborators are mentioned here, all the funding resources, all the fantastic alumni who worked on this work, as well as my current lab. And I'd be happy to answer further questions if you may have some.
Thank you very much, Bart. It was really lots of information.
Maybe
and
too much, huh?
We have learned a lot. I worked with circadian clock during my PhD, but it was 20 years ago. So I forgot some things, but now you just made me to remember some things about circadian clock. It is really amazing. It's really amazing what you have found. So anybody has a question? Questions?
You already asked a lot, but I'd be happy to answer more.
In the samples you have studied of that mutant, so have you looked for changes in the microbiota?
No,
in the gut
no.
samples?
Actually, you know, one of the hopes that we had was to also look at gene expression, because somehow actually, you know, sometimes you actually carry over some of gene expression from bacteria to actually look whether there's any change, but no, we have not done that. And you know, these lines are available for free, right? So if this is actually of interest to you, you could easily get that line. You know, some other labs have already basically recapitulated the phenotype. For example, Sebastian Kadener in Brandeis, who's really a circadian biology expert, has also looked at this line independently, and so this really works. So if that would be of interest, then you know the resource is available to everyone. So I really encourage you to just get your hands on this line and sort of study this, because I'm sure there's very interesting phenotypes there as well in the microbiota. This is definitely a cool question. Yeah.
Great. When you were talking about the cell atlas in Drosophila, you told that insects, they are good for studying because you can separate the body from the head. Why you cannot grind everything together? Why to separate bodies and head?
That's a good question. So far, basically, the idea is to simply to make sure that basically neuronal cells can be mapped to the head versus the body, because sometimes, you know, it's sort of impossible to know whether, you know, if you take the everything together, where these neurons are specifically coming from. So at least it provides you some level of stratification. But in principle, you could do the entire insect if you'd like. It's just that I think it's sort of convenient to at least know, especially for neurons, whether they're basically likely part of the brain or whether they're sort of interspersed actually with individual tissues in the body. So it's more of a practical arrangement. And also, I think, you know, the overall procedure of extraction of cells, and specifically nuclei, which we used for the fly cell atlas, might be slightly different. So I'll be curious also to hear Robert and Max whether they actually see differences in the overall nuclei extraction efficiency in the body versus the head. So I think we want to make sure that they're equally well represented, and so that's another practical reason, basically. Yeah.
Okay. Okay. And when you were talking about the DGRP lines, the Drosophila DGRP lines, you mentioned that they are independent. I mean,
yeah.
If I remember well, they were constructed by one single female, wasn't it?
Yeah,
so how
one single female per line, right?
Per line. Okay, so there were 140 single females giving origin to the lines.
So Trudy started actually with 1,000, you know, fertilized females basically, and separated them all 1,000, and then through sibling mating, right, started actually the construction of those lines. Now, of those 1,000 lines, basically, she lost roughly 800 in the inbreeding process, right? And I sort of failed to explain why we only looked at 140, because it's our sort of belief and sort of based on observations also that at least 60 are still not that phenotypically robust, right? So there's 60 that are still quite weak. They're more difficult to maintain. And so often these 60 are basically not included in phenotypic studies because they're sort of weak in general, right? So we really have at the moment 140 lines, which are generally considered as very robust and independent, right? And that's why actually the study of mitochondrial DNA variation was relevant, because each mother provided its own mitochondrial DNA, right? In our case, basically, you know, Louis, with the mosquito riddles that we're generating, there should be only 4 mitochondrial haplotypes in principle, right? Because there's only 4 lines that we started off with, right? So that makes, of course, mitochondrial genotype phenotype association studies very difficult, because you only have 4, right? So that's
can we say that the mosquitoes lines we are constructing here in Brazil, they are independent as well? Because we took 4 populations in the wild, and then after constructing Brazil population, that was the result of intercrosses between these populations. I took 1 female per line as well. I started doing 200 lines, and I lost lines due to inbreeding effects, but they were, the origin for each line was 1 single female as well.
Yes, but we only started actually from 4, right? I mean, in a way, what you would have done is basically for each location, you would have had 250 individual females, right? And then actually built those lines based on that. What we did, of course, well, what you did, Louis, I mean, let's give credit to all the work you've done, is basically sort of mix all those lines initially together, right?
Yes.
So that actually makes them truly recombinant inbred lines, and not sort of, which is fine. It's just that basically the DGRP is different in that way, because it's basically, they're all independent. They were never crossed together, right? Yeah.
I see. Okay.
Yeah. Which complicates genophenotype studies, of course, a little bit more, because you only have so many alleles in your population. In the DGRP, we clearly have, you know, sort of 140 alleles per gene, so to speak, right? In your case, in principle, we should only have 4, you know, in principle, well, 6, I guess there's 4, so that, yeah. So we have 8 alleles basically that will segregate per gene, right? In a diploid sort of context, right?
I see. Great.
And those 8 could, of course, have different combinations. So there's, you know, we can go from there. And so that actually makes genophenotype studies, of course, possible. But the extent of phenotypic variation might be a little bit more limited because of that, which is fine. There's not a problem, because the entire all mouse panels are actually based on this principle, right? They're all recombinant inbred lines, right? And still, they've yielded a ton of interesting information with respect to phenotypic behavior, you know.
Yeah, but because they were isogenic, the mouse, they were isogenic.
Yeah, that of course, but let's make sure to understand they're isogenic and not quite, right? I mean, they also have heterozygosity that is remaining, right? So we call this, of course, isogenic lines, but you know, for 95% this is the case, but there's still actually 5% or so, 5 to 10% of heterozygous sites in those mice, right? Certain sites just refuse to become homozygous for one reason or the next, which I think is already a very interesting genetic conundrum by itself. Yeah. And in mosquitoes, it's particularly abundant, right?
Abundant, yeah, that's really amazing.
Yeah, exactly. Yeah. Mm-hmm.
Great. Well done. Thank you very much.
You're more than welcome. Any other questions? I guess not. I look at the. I see a question actually on the chat. Is there plans for make the scope data integrated more thoroughly, like in digital fly single cell from related to the arts lab? Yeah. A great question also. Can we actually integrate the single cell data with overall morphology? And I think, you know, at least the best I can tell you is, of course, that now there's a lot of interest to do also spatial transcriptomics. So I think that's probably actually going in that direction. I already know, for example, that Stain Arts is also working on that. We are not doing that. I prefer to sort of move to a more obscure yet very interesting insect mosquito. But indeed, I think sort of spatial aspects, which might be related to morphology and single cell transcriptomes will actually increasingly be linked. And that is, of course, sort of the new frontier that is upcoming as well, which I think is also very exciting, right? So this was Alison who was asking this question. So I hope that answered the question a little bit. Okay.
Right. So have a good night. Thank you very much.
Thank you, Bart.
Thank you. It was a pleasure.
Thank you.
And if anybody would have any questions or be interested in any of the resources that I talked about, then just shoot me an email, okay?
Yeah, great.
All right.
Congratulations.
Okay.
Thank you.
Bye-bye.
Ciao.
Bye.
Ciao.
Tchau. Tchau, pessoal.
Ciao.
Ciao.
Tchau, boa tarde. Valeu.
Porra.